A biological safety cabinet, also known as biosafety cabinet or BSC, is an enclosed workspace with a ventilated hood that is designed to contain pathogenic microorganisms during microbiological processes. Biosafety cabinets are often used for procedures involving highly infectious or pathogenic agents including bacteria, viruses, and fungi. It is also designed to both protect laboratory workers or personnel like students, instructors, and the surrounding environment from pathogens or from exposure to biohazards and cross-contamination during routine procedures. Biosafety cabinets are only used for certain risk group organisms and for processes that might result in aerosol formation. BSCs are an essential part of biosafety as they minimize the formation of aerosol. Besides, most BSCs also function to sterilize biological materials that are kept inside the cabinets. The purpose of biosafety cabinet in a microbiology laboratory is to provide a safe, contained environment for handling potentially hazardous biological materials. So in addition to these primary functions, biosafety cabinets are also used to control and contain aerosols and splashes that might be generated during certain laboratory procedures. So this is particularly important when working with infectious agents that can be transmitted via the airborne route. Actually, there are three types of biosafety cabinets: Class 1, Class 2, and Class 3. Class 1 provides protection for personnel and the environment but not the sample. Class 2 provides protection for all three: the worker, the environment, and the sample. While the Class 3 provides the highest level of protection and is used with high-risk pathogens. So as we can see, this is a Class 2 biosafety cabinet. The primary purpose of this is to protect the laboratory personnel, the sample, and the environment from the pathogenic microorganisms as aerosols might be formed during the processing of such microorganisms.
Here are the parts of Class 2 biosafety cabinets. First, work area or work zone. This is the enclosed space where the operator performs tasks involving biological materials. It's designed to maintain a sterile environment and protect both the operator and the samples. Second, front opening or sash. The transparent front barrier that can be raised or lowered. It protects the work area from external contaminants and helps control airflow within the cabinet. Third, HEPA filters or high-efficiency particulate air filters. HEPA filters capture airborne particles and microorganisms, ensuring that the air entering the cabinet is clean and free of contaminants. Fourth is inflow vent. It is used to draw external air into the cabinet, creating inward airflow that prevents airborne contaminants from escaping the work area. Fifth is downflow filter or optional. It is used some Class 2 biosafety cabinets that have a downflow filter above the work surface. It provides traditional filtration to ensure that the air directed into the work zone is sterile. 6 is exhaust system. It is used to remove contaminated air from the cabinet and either directs it through HEPA filters or exhaust it safely outside the laboratory.
UV germicidal lamp. It is optional to use. This lamp emits ultraviolet or UV light, which helps sterilize the working area and equipment. It is used as an additional decontamination measure. Work surface. The flat surface where samples and equipment are placed during operations. It is usually made of stainless steel for easy cleaning and decontamination. Control panel allows the operator to control various functions of the biosafety cabinet, such as airflow velocity, UV lamp operation, and lighting intensity. Alarm system alert the operator to any issues or malfunctions within the cabinet, such as airflow disruptions or filter saturation, ensuring safety and performance. Uses of Class 2 biosafety cabinets in microbiological research and cell culture. BSCs are extensively used in microbiology laboratories for various tasks such as culturing bacteria, viruses, fungi, and other microorganisms, as well as handling biological samples and conducting experiments. They provide a controlled environment for cell culture work, including cell line maintenance, cell-based assay, tissue culture, and various propagation. Overall, Class 2 biosafety cabinets are indispensable tools in laboratories where containment, protection, and sterility are paramount for working with hazardous biological material safely and effectively.
Biosafety cabinets, or BSCs, are essential for handling biological materials safely in laboratories. They provide personnel and product protection from biohazardous materials by creating a unidirectional airflow that moves from the cabinet interior to the exterior. To ensure safe usage, follow these guidelines. First, wear appropriate personal protective equipment, or PPE, including lab coats, gloves, and eye protection. Second, disinfect all interior surfaces of the biosafety cabinet with an appropriate disinfectant before and after use. Next, place materials and equipment logically inside the cabinet, ensuring they do not block grills and are at least 4 inches from the front grill of the working surface. Fourth, limit movement in and out of the BSCs to minimize airflows disruptions. Use a checklist to ensure all necessary items are in the cabinet before starting a procedure. Manipulate materials after allowing the hood environment to stabilize for 1 minute after inserting arms into the hood. Next, read the operator's manual and follow all manufacturer's recommendations. Ensure the cabinet is operating correctly before beginning work. Lastly, restrict use of the cabinet to one person at a time, unless a risk assessment and consultation with the biosafety officer allows for 2 persons to work simultaneously. By adhering to these guidelines, you can help maintain a safe laboratory environment and protect yourself and others from potential biohazards.
We're gonna tackle the step-by-step on how to operate the biosafety cabinet. First is preparation. Put on the appropriate personal protective equipment such as gloves, lab coat, and safety goggles. Ensure that the biosafety cabinet is clean and free of any clutter or unnecessary items. Plug in the power cord of the biosafety cabinet to a properly grounded electrical outlet. Second is power on. Locate the control panel of the biosafety cabinet. Turn on the power switch to activate the cabinet. Third is set airflow velocity. Use the control panel to set the desired airflow velocity, typically measured in linear feet per minute. The recommended airflow velocity for a Class 2 biosafety cabinet is usually between in the 75 and 100 LFPM. Fourth is turn on fluorescent light. Locate the switch or button for the fluorescent light on the control panel. Turn on the fluorescent light to provide illumination within the cabinet. Fifth is turn on UV germicidal lamp. This is an optional. If your biosafety cabinet is equipped with a UV germicidal lamp, you can turn it on using the control panel. The UV lamp helps sterilize the work area and equipment. Only use it when the cabinet is not in use, as UV light can be harmful to the operator.
Next is open the front opening or sash. Raise the transparent front barrier or sash to the desired height. Sash acts as a protective barrier and helps control airflow with the cabinet. Position it at the height that provides adequate protection for the operator and prevents contamination from the entering work area. And the next one is start the exhaust system. Ensure that the exhaust system is properly functioning and directing the air either through HEPA filtration or safely outside the laboratory. The exhaust system is responsible for removing contaminated air from the cabinet. Nextly, perform work in the work area. Place your sample and equipment on the work surface inside the cabinet. Perform your tasks involving biological materials while keeping them within the work area. Avoid obstructing the airflow within the cabinet and reaching over the sash. Close the front opening or sash. Once you have completed your work, lower the sash to its closed position. This helps maintain a sterile environment within the cabinet and prevents contamination from entering. The next one is decontamination. After closing the sash, initiate the decontamination process if required. Wipe down the work surface and equipment with an appropriate disinfectant following the laboratory's standard operating procedure. Lastly, power off. When finished using the biosafety cabinet, turn off the power switch on the control panel. Unplug the power cord from the electrical outlet. Ensure that the cabinet is left clean and free of any biological materials or debris.